Hello.
Hello, fellow scientists. Hello, doctors.
Chef?
Welcome, farmers. Take a seat. Hi, humanity.
Hi. Why are we here?
Because you have been abusing antibiotics for far too long, you need to know about the consequences of your actions. You can see there, antimicrobial resistance has been known to mankind ever since the first antibiotics were discovered in 1928. Currently, at least 700,000 people die each year due to drug-resistant bacteria, and by 2050 the number is predicted to rise up to 10 million. Antimicrobial resistance is also a threat to the global economy. Right now, according to a CDC study, the estimated cost in the UK to treat infections caused by 6 multi-drug resistant bacteria frequently found in healthcare is more than $4.6 billion annually. According to WHO, by 2030, antibiotic resistance could force up to 24 million people to extreme poverty and could lead to a financial crisis more catastrophic than the one in 2008-2009. Antibiotic resistance genes have even been found in samples taken from brown bears in Sweden, proving that almost immediately after they were introduced to the public in the 1950s, they penetrated even the remotest Swedish forests. Meanwhile, in China, one of the currently worst areas for antibiotic abuse, the annual prescription of antibiotics, including both clinical and veterinary treatment, is approaching 140 grams per person.
So I'm just doing what law says I can do. Why is it my concern?
But you're using way too much, and even one resistant cell can cause a problem not just to your farm, but to the environment surrounding it. If you look back at the presentation, worldwide, it is estimated that 66% of all antibiotics are used in farm animals and not people. Much of this use is routine and enables farm animals to be kept in poor conditions where disease spreads easily. An inherent consequence of this, antibiotic resistance, rises as a result of natural selection. Due to normal genetic variation in bacteria populations, individual organisms may carry mutations that render antibiotics ineffective. Not only that, but in the presence of antibiotics, advantageous mutations can also be transferred via vertical or horizontal transfer. In vertical gene transfer, the transfer of genetic material is from parent to offspring, or from one cell to its daughter cells. Horizontal transfer, on the other hand, is the movement of genetic information between organisms in one of three ways: transformation, conjugation, and transduction. Once transferred, the genes and pathogens continue to evolve, often resulting in bacteria with greater resistance. Giving so many antibiotics to farm animals can result in a rapid increase and spread of antibiotic resistance. We proved this by running an experiment where we first applied lethal doses of antibiotic to cells to kill them, proving they didn't carry any resistance genes. Afterwards, we applied sublethal doses at which the cells were able to grow under selective pressure. This allowed them to mutate and develop resistance genes to the antibiotic and survive being placed in concentrations far beyond that of normally prescribed antibiotics.
I agree with you, but we can't exactly stop using antibiotics, and we definitely can't take samples from every living thing that takes them.
We can take samples from the environment and check what is being released there, which is where we come in. We used wastewater samples, some provided to us by the Department of Hydrobiology in Sofia University, some collected in nearby farms, and some given to us as a part of a collaboration. The samples were taken safely using proper equipment. Then we ran full metagenomic tests, which means we sequenced and tested all the DNA found in these samples. We were looking for antibiotic resistance genes that may have occurred because of the excess use of antibiotics in the area.
That sounds great, but can you actually do that?
Yes, and we did. First, we isolated the DNA in the samples. Then, using PCR and the primers we developed, made specifically to amplify variable gene cassettes of class 1 integrons, we amplified the region we believed carried the antibiotic resistance gene. Then, we also developed a novel T-cloning vector that allows efficient cloning of integron gene cassettes. Then, using the TA cloning method, we combined the two and used them to transform competent Escherichia coli cells.
Okay, but what are you gonna do with the data?
We plan to offer a service where hospitals and farms can send us samples to analyze, and we send the results back to them. We will be running full metagenomic tests on the samples and using the data to figure out if there are any already known or new genes that lead to antibiotic resistance. This way, doctors and vets can have the full information they need to choose the right broad-spectrum antibiotic in the beginning phases of the treatment before conducting an antibiogram. This will vastly improve treatment quality and slow the occurrence of antibiotic resistance.
But isn't that dangerous?
No, the samples are taken from the environment and the only thing amplified is a small region only found in bacteria. That means that no private data is analyzed or stored. We also thought about the risk of one of our genes escaping, which is why we developed an orthogonal system which allowed us to re-engineer the Amber stop codon UAG. That transformed it into a codon responsible for the incorporation of a normal amino acid, which is often found in antibiotic resistance proteins. That ensured that if our genes do get out, there is no way for them to be translated into normal bacteria.
Hello, I'm Teddy, the team leader. Thank you for watching. This was a short film made by Jane Boguer to show the major problem that is antibiotic resistance. But now, let's talk more about our project.
First, we received 1 gigabase sequencing information for a clinical carbapenem-resistant strain, Klebsiella pneumonia, from BGI Genomics. The resulting raw read pairs were uploaded to the Galaxy Online platform you can see here. We investigated the resistome of this strain using the tool Abricate, and all localized antibiotic resistant determinants are shown in the table below. We designed primers that can amplify the variable gene cassettes of class 1 integrons and successfully applied them to a control strain of Pseudomonas aeruginosa. Because of the nature of our project, we needed to figure out a way to clone our unsequenced constructs without exposing them to restriction enzymes, which is why we chose TA cloning. In order to optimize the cloning of the integron gene cassettes, we developed a novel T-cloning vector. We designed 3 primer pairs that can amplify the QR-DR region of the GA gene in Escherichia coli, as well as a reporter vector that contains the remaining part of the GA coding sequence. We created a novel cloning-based system that can be used for monitoring the formation of unproductive chimera and artifacts between highly similar DNA sequences and PCR amplifications. We needed this tool to see if the PCR amplification step of the QR-DR regions found in complex samples can lead to chimera formation that has the potential to bias our results. This system consists of 2 parts that were submitted to the registry. They both consist of MRFP1 coding sequence, but 2 tyrosine codons in them were mutated to Amber TAG stop codons. We would also like to present you our model. Everything started with the simple question: how does antibiotic resistance occur and spread? We wanted to show everyone what can happen if one uses the wrong dosage and/or time course for antibiotic treatment. To make this as obvious and impressive as possible, we decided to build a real-world model that will grow an antibiotic-resistant bacterial strain. We use standard laboratory Escherichia coli KRX cells that are sensitive to the antibiotic rifampicin. First, we prepared liquid LB medium with gradually increasing concentrations of rifampicin. We found that everything above 2.5 micrograms per milliliter is lethal for the cells. No growth is observed, even upon prolonged cultivation times. Meanwhile, in concentrations of 0.5 to 2 micrograms per milliliter, the cells were able to grow, but the present antibiotic had a selective pressure over them. We took aliquot from the 2 micrograms per milliliter condition and we placed it in media with rifampicin concentrations of 2.25 to 7.5 microgram per milliliter. Despite that the cells were not able to grow in these conditions before, now we observed growth everywhere. This is highly concerning since it clearly illustrates how quickly antibiotic resistance can occur even in a non-virulent and non-hypermutable strain if an insufficient dosage of the medicament is applied. To demonstrate that even continuous increase in the antibiotic dosage will not help in such case, we built a mini bioreactor that utilizes a peristaltic pump that increases the rifampicin concentrations with time. After 24 hours, we reached concentrations of about 30 micrograms per milliliter and bacterial growth was still observable. Finally, to prove that the Escherichia coli cells developed resistance against rifampicin, we made antibiograms with rifampicin discs, using all types of cells that were able to grow in media with more than 2 micrograms per milliliter of antibiotic. They all showed no zone of growth inhibition around the discs. In contrast, the initial cells showed a clear and nicely visible zone of growth inhibition. As part of this year's contributions, we also optimized protocols for colorful agar. We tested 2 different compounds, titanium dioxide and activated charcoal, that led to white and black agar respectively. Next, we assembled different expression constructs with chromoproteins to see if they show improved visibility compared to normal LB agar. You can see some representative results on the following pictures. The overall conclusion is that red chromoproteins are best observed on white media, while the yellow ones show optimal visibility on the black agar. Lastly, this year, as we had a number of engineers on our team, we tried to develop a cell growth kinetics measurement system. Our engineering team used a Raspberry Camera V2 to capture the images of petri dishes, which is lit by our own 3D printed reflector. Using 3D printing, we also made a barrel that can hold 5 90mm petri dishes. It is powered by an EMA 17 stepper motor, which is also connected to the Raspberry Pi using a stepper driver. While we are still having some trouble with the lighting, you can see some preliminary testing pictures here. Finally, it was also very important for us to talk to other scientists about the problem that is antibiotic resistance and exchange ideas. To do this, we took part in several events such as the International Microbiome Day, organized by our key sponsors, Ridacom and the Sofia Science Festival. We also had the pleasure of visiting the Department of Hydrobiology in the Faculty of Biology of Sofia University, where they showed us around the lab and introduced us to their work. Thank you for your attention.